Chapter 2: Human Reproduction. As you are aware, humans are sexually reproducing viviparous. The reproductive events in humans include formation of gametes, gametogenesis, sperms in males and ovum in females, transfer of sperms into the female genital tract, insemination and fusion of male and female gamete, fertilization leading to the formation of zygote. This is followed by the formation and development of blastocysts and attachment to the uterine wall, implantation, embryonic development, gestation, and the delivery of the baby per duration. You have learned that these reproductive events occur after puberty. There are remarkable differences between the reproductive events in the male and in females. For example, sperm formation continues even in old men, but the formation of the ovum ceases in women around the age of 50 years. Let us examine the male and female reproductive system in humans. The male reproductive system. The male reproductive system is located in the pelvis region. It includes a pair of testes along with the accessory ducts, glands, and external genitalia. The testes are situated outside the abdominal cavity within a pouch called scrotum. The scrotum helps in maintaining the low temperature of testes 2 to 2.5 degrees Celsius, lower than the normal internal body temperature necessary for spermatogenesis. In adults, each testis is ovule, oval in shape with a length of about 4 to 5 centimeter and a width of about 2 to 3 centimeter. The testis is covered by the dense covering. Each testis has about 250 compartments called testicular lobules. Figure 2.1b. Each lobule contains 1 to 3 highly coiled seminiferous tubules in which sperms are produced. Each seminiferous tubule is lined on its inside by two types of cells called male germ cell, spermatogonia, and Sertoli cells. The male germ cell undergo meiotic division, finally leading to sperm formation, while Sertoli cells provide nutrition to the germ cell. The regions outside the seminiferous tubule called the interstitial space contain small blood vessels and interstitial cells or Leydig cells. Leydig cells synthesize and secrete testicular hormones called the androgens. Other immunologically competent cells are also present. The male sex accessory ducts include testis, vasa differentia, epididymis, and vas deferens. The seminiferous tubule of the testis open into the vasa efferentia through rete testis. The vasa efferentia leave the testis and open into the epididymis located the posterior surface of each testis. The epididymis leads to vasa deferens that ascends to the abdomen and loops over the urinary bladder. It receives a duct from a seminal vesicle, opens into the urethra as the ejaculated duct. These ducts store and transport the sperm from the testes to the outside through urethra. The urethra originates from the urinary bladder and extends through the penis to its external opening called urethral meatus. The penis is the male external genitalia. It is made up of special tissue that helps in erection of the penis to facilitate insemination. The enlarged end of the penis called the glans penis is covered by the loose fold of skin foreskin. The male accessory gland include paired seminal vesicle, a prostate, and paired bulbourethral gland. Secretion of these glands constitute the seminal plasma, which is rich in fructose, calcium, and certain enzymes. The secretion of bulbourethral glands also helps in lubrication of the penis. The female reproductive system. The female reproductive system consists of a pair of ovaries along with a pair of oviducts, uterus, cervix, vagina, and external genitalia, located in pelvic region. These parts of the system, along with a pair of the mammary gland, are integrated structurally and functionally to support the process of the ovulation, fertilization, pregnancy, birth, and child care. Ovaries are the primary female sex organs that produce the female gamete ovum and several steroid hormones or ovarian hormones. The ovaries are located one on each side of the lower abdomen. Each ovary is about 2 to 4 centimeter in length and is connected to the pelvic wall and uterus by ligaments. Each ovary is covered by a thin epithelium which encloses the ovarian stroma. The stroma is divided into two zones, a peripheral cortex and the inner medulla. The oviducts, fallopian tube, uterus, and vagina constitute the female accessory ducts. Each fallopian tube is about 10 to 12 centimeter long and extend from the periphery of each ovary to the uterus. The part closer to the ovary is the funnel-shaped infundibulum. The edges of infundibulum poses a finger-like projection called fimbriae, which helps in collection of the ovum after ovulation. The infundibulum leads to a wider part of the oviduct called ampulla. The last part of oviduct isthmus has a narrow lumen and it joins the uterus. The uterus is a single and it also called womb. The shape of uterus is like a inverted pear. It is supported by ligaments attached to a pelvic wall. The uterus opens into vagina through a narrow cervix. The cavity of the cervix is called cervical canal, which along with vagina forms the birth canal. The wall of the uterus has three layers of tissue: the external thin membranous perimetrium, middle thick layer of smooth muscle myometrium, and inner glandular layer called endometrium that lines the uterine cavity. The endometrium undergoes cyclical changes during menstrual cycle, while the myometrium exhibits strong contraction during the delivery of the baby. The female external genitalia includes mons pubis, labia majora, labia minora, hymen, and clitoris. Mons pubis is a cushion of fatty tissue covered by skin and pubic hair. The labia majora are fleshy folds of tissue which extended extend down from the mons pubis and surround the vaginal opening. The labia majora are paired folds of tissue. Under the labia majora, the opening of the vagina is often covered partially by a membrane called hymen. The clitoris is a tiny finger-like structure which lies at the upper junction of the two labia minora above the urethral opening. The hymen is often torn during the first coitus intercourse. However, it can also be broken in some sports, like also broken by a sudden fall or jolt, insertion of vaginal tampon, active participation in some sports like horseback riding, cycling, etc. In some women, the hymen persists even after coitus. In fact, that presence or absence hymen is not a reliable indicator of virginity or sexual experience. A functional mammary gland A functional mammary gland is characteristic of all female mammals. The mammary gland are paired structured breasts that contain glandular tissues and variable amount of fat. The glandular tissue of each breast is divided into 15 to 20 mammary lobes containing clusters of cells called alveoli. The cells of alveoli secrete milk which is stored in the cavities lumens of alveoli. Lumens of alveoli. The alveoli open into mammary tubules. The tubules of each lobe join to form mammary duct. Several mammary ducts join to form a wider mammary ampulla which is connected to lactiferous through which milk is sucked out. Gametogenesis. The primary sex organ, the testes in the male and the ovaries in the female, produce a gametes, sperms, and the ovule respectively. By the process called gametogenesis in testes, the immature male germ cells spermatogonia produce sperms by spermatogenesis that begins at puberty. The spermatogonia presents on the inside wall of seminiferous tubule multiply by meiotic division and increase in numbers. Each spermatogonia is diploid and contain 46 chromosomes. Some of spermatogonia called primary spermatocyte periodically undergo meiosis. A primary spermatocyte completes the first meiotic reduction division, leading to the formation of two equal haploid cells called secondary spermatocytes, which have only 23 chromosomes each. The secondary spermatocyte undergoes the second meiotic division to produce four equal haploid spermatids. What would be the number of the chromosomes in the spermatids? The spermatids are transformed into the spermatozoa sperms by the process called spermiogenesis. After spermiogenesis, sperm heads become embedded in the Sertoli cells and are finally released from the seminiferous tubules by the process of the called spermiation. Spermatogenesis starts at the age of the puberty due to the significant increase in the secretion of the gonadotropin releasing hormone GnRH. This, if you recall, is the hypothalamic hormone. The increased level of GnRH then acts at the anterior pituitary gland and stimulates secretion of two gonadotropin luteinizing hormone and follicular stimulating hormone FSH. LH and the LH acts at the Leydig cell and stimulates synthesis and secretion of androgens. Androgens in turn stimulate the process of spermatogenesis. FSH act on the Sertoli cells and stimulates secretion of some factors which help in the process of spermiogenesis. Let us examine the structure of sperm. It is microscope structure composed of a head, neck, a middle piece, and a tail. A plasma membrane envelops the whole body of a sperm. The sperm head contains an elongated haploid nucleus, the anterior portion of which is covered by a cap-like structure acrosome. The acrosome is filled with enzymes that help fertilization of ovum. The middle piece process possesses numerous mitochondria which produce energy for the movement of the tail that facilitate sperm motility. Essential for fertilization. The human man ejaculates about 200 to 300 million sperms during a coitus of the which for normal fertility. At least 60% sperms must have normal shape and size, and at least 40% of them must show vigorous motility. Sperm released from the seminiferous tubule and separate are transported by the accessory ducts, secretion of epididymis, vas deferens, seminal vesicle, and prostate are essential for the maturation and motility of sperms. The seminal plasma, along with the sperms, constitute the semen. The function of male sex accessory ducts and glands are maintained by the testicular hormone androgens. The process formation of a mature female gamete is called oogenesis, which is markedly different from spermatogenesis. Oogenesis is initiated during the embryonic development stage when a couple of million gamete mother cell oogonia are formed within each fetal ovary. No more oogonia are formed and added after birth. These cells start division and enter into the prophase 1 of the meiotic division and get temporarily arrested at the stage called primary oocyte. Each primary oocyte then gets surrounded by a layer of the granulosa cells and is called primary follicle. A large number of these follicles degenerate during the phase from birth to puberty. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovary. The primary follicle gets surrounded by more layers of the granulosa cells in the new theca and are called secondary follicles. The secondary follicle soon transform into a tertiary follicle, which is characterized by the fluid-filled cavity called antrum. The theca layer is organized into an inner theca interna and outer theca externa. It is important to draw your attention that it is at this stage that the primary oocyte within the tertiary follicle grows in size and completes its first meiotic division. It is an unequal division resulting in the formation of the large haploid secondary oocyte and a tiny first polar body. The secondary oocyte retains bulk of the nutrient rich cytoplasm of the primary oocyte. Can you think of any advantage for this? Does the first polar body bond out of first meiotic division divide further or degenerate? At present, we are not very certain about this. The tertiary follicle further changes into the mature follicle or graphene follicle. The secondary oocyte forms a new membrane called zona pellucida surrounding it. The graphene follicle now ruptures to release the secondary oocyte ovum from the ovary by the process called the ovulation. Can you identify major differences between the spermatogenesis and oogenesis? A diagrammatic representation of spermatogenesis and oogenesis is given below, figure 2.8. Menstrual cycle 2.4. Menstrual cycle. The reproductive cycle in the female primates, example monkeys, apes, and human beings, is called menstrual cycle. The first menstruation begins at the puberty and is called menarche. In human females, menstruation is repeated at an average interval of about 28 to 29 days, and the cycle of the event starting from one menstruation till the next one is called the menstrual cycle. One ovum is released ovulation during the middle of each menstrual cycle. The major events of the menstrual cycle are shown in the figure 2.9. The cycle starts with the menstrual phase and when the results due to breakdown endometrial lining of the uterus and its blood vessels, which forms liquid that comes out through vagina. Menstruation only occur if released ovum is not fertilized. Lack of menstruation may be indicative of pregnancy. However, it may also be caused due to some other underlying causes like stress, poor health, etc. The menstrual phase is followed by the follicular phase. During this phase, the primary follicles in the ovary grow to become a fully mature Graafian follicle, and simultaneously the endometrium of uterus regenerates through proliferation. These changes in the ovary and uterus are induced by the changes in the levels of pituitary and ovarian hormone. The secretion of gonadotropin, LH, and FSH increase gradually during the follicular phase and stimulates the follicular development as well as secretion of the estrogens by growing follicle. Both LH and FSH attain a peak level in the middle of cycle. About 14th day, rapid secretion of the LH leading to its maximum level during the mid-cycle called LH surge, induce rupture of Graafian follicle and thereby the release of ovum ovulation. The ovulatory phase is followed by the luteal phase, and during which the remaining part of the Graafian follicle transforms as the corpus luteum. The corpus luteum secretes large amount of progesterone, which is essential for the maintenance of the endometrium. Such an endometrium is necessary for implantation of the fertilized ovum and other events of pregnancy. During pregnancy, all events of the menstrual cycle stop and there is a no menstruation. In the absence of fertilization, the corpus luteum degenerates. This causes disintegration of endometrium, leading to the menstruation, marking a new cycle. In human beings, menstrual cycle ceases around 50 years of age. That is termed as menopause. Cyclic menstruation is an indicator of normal reproductive phase and extend between menarche and menopause. 2.5 Fertilization and implantation. During copulation, coitus semen is released by the penis into the vagina. Insemination, the motile sperm swim rapidly pass through the cervix, enter into the uterus, and finally reach the ampullary region of the fallopian tube. The ovum released by the ovary is also transported to the ampullary region where fertilization takes place. Fertilization can only occur if the ovum and sperms are transported simultaneously to the ampullary region. This is the reason why not all copulation lead to fertilization and pregnancy. The process of fusion of sperms with an ovum is called fertilization. During fertilization, a sperm comes in contact with the zona pellucida layer of the ovum, figure 2.10, and induces changes in the membrane that blocks the entry of additional sperms. Thus, it ensures that only one sperm can fertilize an ovum. The secretion of the acrosome help in the sperm enter into the cytoplasm of the ovum through the zona pellucida and the plasma membrane. This induces the completion of the meiotic division of the secondary oocyte. The second meiotic division is unequal and results in the formation of the second polar body and a haploid ovum oocyte. Soon the haploid nucleus of the sperms and that of the ovum fuse together to form a diploid zygote. How many chromosomes will be there in the zygote? One has to remember that the sex of the baby has been decided at this stage itself. Let us see how. As you know, the chromosome pattern in the human female is XX and that in the male XY. Therefore, all the haploid gametes who are produced by the female have the sex chromosome X, where in the male gametes sperms the X sex chromosome could be either X or Y. Hence, 50% of sperms can carry X, and while the 50% of carry the Y. After fusion of the male and female gametes, the zygote would carry either XX or XY, depending on whether the sperm carrying X or XY fertilize the ovum. The zygote carrying XX would develop into a female baby, and XY would form a male. You will learn more about chromosomal patterns in chapter 5. That is why scientifically it's correct to say that the sex of baby determined by the father and not by the mother. The meiotic division starts as the zygote moves through the isthmus of oviduct called cleavage towards uterus and forms 2, 4, 8, 16 daughter cells called blastomeres. The embryo with 8 to 16 blastomeres is called morula. The morula continues to divide, transform into blastocyst as it moves further into the uterus. The blastomeres is the blastocyst are arranged into an outer layer called trophoblast, an inner group of cells attached to trophoblast called the inner cell mass. The trophoblast layer then gets attached to the endometrium, and the inner cell mass gets differentiated as the embryo after attachment. The uterine cells divide rapidly and covers the blastocyst as a result. The blastocyst become embedded in the endometrium of the uterus. This is called implantation and leads to pregnancy. Pregnancy and embryonic development. After implantation, finger-like projection appear on the trophoblast called chorionic villi, which are supported by the uterine tissue and the maternal blood. The chorionic villi and the uterine tissue become interdigited with each other and jointly form a structural and functional unit between the developing embryo, fetus, and maternal body called placenta. The placenta facilitates the supply of oxygen and nutrients to the embryo and also removal of carbon dioxide and excretory waste material produced by the embryo. The placenta is connected to the embryo through the umbilical cord, which helps in the transport of the substances to and from the embryo. The placenta also acts as endocrine tissue and produces several hormones like human chorionic gonadotropin (HCG), human several HCG, human placental lactogen HPL, estrogen, progesterone, etc. In the later phase of pregnancy, a hormone called relaxin is also secreted by the ovary. Let us remember the HCG, HPL, and relaxin are produced in women only during pregnancy. In addition, during pregnancy, the levels of the hormones like estrogen, progesterone, cortisol, prolactin, thyroxine, etc. are increased several folds in the maternal blood. Increased production of these hormones is essential for supporting the fetal growth, metabolic changes in the mother, and maintenance of the pregnancy. Immediately after implantation, the inner cell mass embryo differentiates into an outer layer called ectoderm and inner layer called endoderm. A mesoderm soon appears between the ectoderm and the endoderm. These three layers give rise to the all tissue organs in adults. It needs to be mentioned here that the inner cell mass contains certain cells called stem cells, which have the potency to give rise to all tissue and organs. What are the major features of embryonic development at the various months of the pregnancy? The human pregnancy lasts 9 months. Do you know for how many months pregnancy lasts in dogs, elephants, cats? Find out. In human beings, after 1 month of pregnancy, the embryo's heart is formed. The first sign of growing fetus may be noticed by listening to the heart sound carefully through the stethoscope. By the end of the second month of the pregnancy, the fetus develops limbs and digits. By the end of 12 weeks, first trimester, most of the major organ systems are developed. The first movements of the fetus and appearance of the hair on the head are usually observed during the fifth month. By the end of the about 24 weeks and the second trimester, the body is covered by with fine hair. eyelids separated and eyelashes are formed by the end of the 9 months of the pregnancy. The fetus is fully developed and is ready for the delivery. Parturition and lactation. The average duration of a human pregnancy is about 9 months, which is called the gestation period. Vigorous contractions of uterus at the end of the pregnancy cause explosion, delivery of the fetus. This process of the delivery of the fetus, childbirth, is called parturition. Parturition is induced by a complex neuroendocrine mechanism. The signals for parturition originate from the fully developed fetus and the placenta, which induces mild uterine contraction called fetal ejection reflex. This triggers release of oxytocin from the maternal pituitary. Oxytocin act on the uterine muscle and causes stronger uterine contraction, which in turn stimulate further secretion of oxytocin. The stimulatory reflex between the uterine contraction and oxytocin secretion and continuous, resulting in the stronger and stronger contraction. This leads to explosion of the baby out of the uterus through the birth canal. Parturition soon after the infant is delivered, the placenta is also expelled out of the uterus. What do you think the doctors inject to induce delivery? The mammary glands of the female undergo differentiation during pregnancy and starts producing milk towards the towards the mammary glands of the female undergo differentiation during pregnancy and start producing milk towards the end of the pregnancy by the process of the called lactation. This helps the mother in the feeding the new born. The milk produced during the initial few days of the lactation is called colostrum, which contains several antibodies absolutely essential to develop resistance for the newborn babies. Breastfeeding during the initial period of infant growth is recommended by the doctors for bringing up a healthy baby. Chapter finished. Thank you.